I became aware of how difficult it is to prepare samples and process biological samples in general during my PhD at Cornell. When people are preparing genomic samples right now, they generally have to go through a series of steps in order to get to the DNA to a point where it can go into further analysis. That generally involves an extraction of the DNA, a purification of that DNA, and then a sizing of the DNA down to the proper sizes for the subsequent analysis. Generally, when people are doing this, they utilize beads and columns technologies, binding technologies that rely on binding affinities to hold on to the DNA, and they have to wash and elute that DNA, dehydrating it and rehydrating it through many cycles, as well as transferring that DNA and sample through many steps. This causes a lot of sample loss and a lot of sample damage. These are things that we can avoid using our system. So our company mission at Inso Bio is to make genomics more accessible, and to do so by building uniquely innovative tools as a front-end processor of biology. The unique aspect of our technology is that we're able to keep the DNA in a solution through the entire processing. And it owes to our name, Inso Bio, keeping it in solution. By doing so, we're actually damaging the DNA less, and our system can extract and purify in the same step, which reduces the amount of time significantly. We're not trying to build a technology that is only useful for a specific type of analysis. What we are trying to build is a tool that can process biological samples for a wide range of genomic analyses. And this includes DNA sequencing for both short-read sequencing as well as long-read sequencing, as well as other types of analysis methods like optical mapping, for example. The micropillar array can hold on to DNA similar to how you can think about a fork holds on to spaghetti. It's purely based off of the long lengths, so it's based off of the size of genomic DNA that is in our cells that it can retain and hold on to that DNA, whereas other molecules such as proteins or lipids or RNA, all of that's washed through our flow cell, and that can be collected separately for multi-omic analysis, or it can be discarded depending on the application. What I believe is that we'll be part of the background. We'll be part of one of those ubiquitous instruments that sits on a lab bench that performs a certain process that everybody sees and recognizes and knows how to use, similar to how a thermocycler is used for PCR. You know, many laboratories have a thermocycler. We wouldn't be able to imagine going through the PCR process without a thermocycler. The underlying core component of our technology is the micropillar array technology. And so what this technology does is two very unique things that no other technology is capable of doing. The first is that it can physically extract DNA. Essentially, it doesn't rely on any surface chemistry or binding chemistry in order to hold on to the DNA when the cells are popped, when they're lysed. The second unique function of our system and this micropillar array is that it can purify the DNA through an inflow process by keeping the DNA in solution without needing to dehydrate and rehydrate that DNA through precipitation steps. My team is mostly comprised of myself and my two co-founders, whom I have immense respect for. My first co-founder is Professor Harold Craighead, is also whom I actually did my PhD with. He was my PhD advisor. And Harold was really the first person to believe in me in a way, and gave me the chance to not only study under him as a PhD student, but also supported me in the journey of the nascent formation of this company. So my second co-founder is Adam Bissoni, and Adam and I had worked together for many years as PhD students and then a few more years as a postdoc. He comes from more of a biological sciences background, and I come from more of an engineering-heavy background. And the way that we've been trained to approach problems actually happens to complement each other very well. I think that our world is set to be changed by genomic technologies and the coming revolution that is brought on through genomic technologies. And similar to the computer revolution that changed our lives and our society, genomics will likely have a similar level of impact. But similar to the computer revolution, it couldn't have all happened if computers weren't made accessible first. And not just for trained computer scientists to use, but for everyone to use. And I think in the same way, that's what we're trying to do at Inso Bio, is make performing genomics and genomic analysis itself more accessible and easier in order to enable the applications, both biological and clinical, of tomorrow.